Good morning, it's June 19th and this is your daily brief in science. Here's everything you need to know. Researchers have made a groundbreaking discovery regarding bogong moths, revealing their ability to navigate using constellations and the Milky Way. This marks them as the first known invertebrates to utilize a stellar compass for long-distance migration. In controlled experiments, the moths demonstrated consistent directional flight patterns aligned with their migration when exposed to simulated spring skies, while reversing their direction under conditions mimicking fall. Lead author David Dreyer's research indicated that when the moths were deprived of magnetic cues and visual stimuli, they became disoriented. However, they regained their directional accuracy when presented with a projection of a starry sky. This study highlights the significance of celestial cues in the navigation strategies of various species, paralleling migratory patterns observed in birds and other animals. The findings emphasize the impressive navigational abilities of these insects and enhance our understanding of animal migration and orientation mechanisms. However, it is crucial to note that the bogong moth species has recently been classified as endangered, experiencing a staggering 99.5% population decline since 2021 due to severe drought conditions and bushfires. The research advocates for the protection of migratory pathways and dark skies, which are essential for the moth's navigation. Moreover, conservation efforts must address the impacts of urbanization and light pollution, which can disorient moths navigating through city environments. These findings could also inform advancements in robotics and drone navigation, as well as conservation strategies for vulnerable species like the bogong moth. Conducted by scientists from Lund University and the Australian National University, the study showcases the migration of approximately 4 million moths each year and was published in the journal Nature. The FDA has approved lenacapavir, marketed as Yeztugo, a new drug that offers nearly complete protection against HIV with just 2 injections per year. This innovative treatment emerged from clinical trials known as Purpose 1 and Purpose 2, which demonstrated a 100% reduction in HIV infections among participants compared to traditional daily oral PrEP options. Despite the promise of lenacapavir, current usage of PrEP in the U.S. remains low, with only about 400,000 Americans utilizing any form of preventive treatment. Barriers such as stigma and low awareness contribute to this limited uptake. Additionally, concerns have been raised regarding the potential high cost of Yeztugo, with estimates suggesting a launch price around $25,000 per year, which could restrict access, especially in low-income countries. Gilead, the manufacturer, is committed to improving accessibility through patient assistance programs and partnerships with telehealth services to help reduce costs for eligible patients. However, challenges persist in ensuring equitable distribution, particularly for marginalized populations who may not benefit from the drug without targeted outreach. Public health experts have highlighted that systemic healthcare issues, including funding cuts to HIV programs, could hinder the effective rollout of lenacapavir. Winnie Byanyima, head of UN AIDS, has advocated for a global rollout of this drug, calling it a miracle tool that could revolutionize preventive treatment and potentially lead to the end of AIDS. Healthcare professionals stress the importance of broader access to effective PrEP options, particularly for vulnerable populations like women and marginalized communities. Continuous monitoring and data collection will be crucial post-approval to ensure lenacapavir meets diverse community needs and improves accessibility. While lenacapavir represents a significant advancement in HIV prevention, its ultimate success will depend on overcoming financial, infrastructural, and outreach challenges. A recent breakthrough in paleoanthropology has emerged from Harbin, China, where DNA analysis and proteomic studies have identified an ancient skull known as dragon-man as a representative of the Denisovans, an enigmatic group of ancient humans. This skull, featuring a unique combination of traits, has sparked debate among scientists about whether it should be classified as a distinct species or simply as a Denisovan. The significance of this discovery lies in its potential to reshape our understanding of human evolution in Asia during the Middle Pleistocene. This period was marked by the coexistence and interbreeding of multiple hominin species, including Homo sapiens, Neanderthals, and Denisovans. The research enhances our comprehension of how these groups interacted, with implications for modern human genetics. The findings also allow for more precise comparisons of Denisovans to other hominin specimens, shedding light on their body proportions and adaptations to various climates. Notably, evidence suggests that Denisovans were large and robust, with estimates indicating a lean body mass of around 100 kilograms. The skull's distinctive features, such as a wide, flattish face, large eye sockets, and robust molars, offer a glimpse into the physical appearance of this ancient group. Researchers have uncovered various Denisovan fossils across Asia, confirming their widespread presence and enhancing our understanding of their diversity. Recent studies indicate that mitochondrial DNA recovered from dental plaque links dragon-man to an early Denisovan population that existed between 217,000 and 106,000 years ago. While the findings support the classification of the skull as Denisovan, some scientists advise caution, highlighting the need for more evidence to definitively classify it due to potential contamination concerns. This ongoing debate underscores the complexity of human evolution and the necessity for further research to fully understand the role of Denisovans in our ancestry. Lead researcher Chao Mei Fu remarked that this discovery clears up some mysteries surrounding Denisovans and opens new avenues for future investigations into East Asian fossils. A groundbreaking study published in Nature has unveiled the remarkable adaptability of ancient Homo sapiens, which played a crucial role in their migration from Africa around 50,000 years ago. This research reveals that all non-Africans today are descended from a small group of individuals who successfully ventured into Eurasia after several previous attempts that did not succeed. The study emphasizes that the ecological flexibility of modern humans was vital for their global dispersal, allowing them to thrive in new and often challenging environments. Researchers conducted an analysis of archaeological evidence from sites dating back to between 120,000 and 14,000 years ago. They noted a significant increase in the variety of habitats occupied by humans approximately 70,000 years ago. During this time, humans transitioned from primarily inhabiting savannas and forests to adapting to more difficult environments, including dense rainforests and arid deserts, demonstrating their impressive ecological flexibility. The study suggests that increased cultural exchange and interaction among human groups may have bolstered this ecological adaptability, enabling them to overcome geographic challenges. This finding challenges previous theories that attributed successful migrations to specific technological advancements or enhanced information sharing methods, instead highlighting the importance of ecological flexibility. Key researchers, including Emily Hallett and Eleanor Scarry, assert that this adaptability did not stem from a single evolutionary change, but rather from a complex interplay of factors, such as broader geographical distribution and cultural exchanges. Sari notes that humans' ability to act as ecosystem generalists was a key factor in their survival across diverse environments. Interestingly, the successful migration into Eurasia occurred during less favorable climatic conditions, suggesting that groups migrating after 60,000 years ago had improved adaptability due to their experiences in varied environments. William Banks from the University of Bordeaux points out that while Homo sapiens exhibited this adaptability, other early human ancestors also migrated and established settlements, raising questions about why only Homo sapiens survived long-term. This research provides valuable insights into the preparedness of Homo sapiens for migration, yet it does not clarify why they alone thrived among other early human species. A recent study from researchers at the University of California, San Diego has unveiled a potential new strategy to combat metabolic diseases linked to high-fat diets through time-restricted feeding, or TRF. This approach has been shown to restore the rhythmic activity of the gut microbiome, a crucial factor in maintaining metabolic health. Over an 8-week experimental period, TRF demonstrated protective effects against metabolic dysfunction in mice. Notably, it restored daily rhythms in microbial gene activity that traditional methods failed to detect, providing new insights into gut health. Using metatranscriptomics, the researchers analyzed daily fluctuations in microbial gene expression. They found that TRF not only alters the presence of microbes but also modifies their activity patterns, significantly influencing metabolism. These findings suggest that similar enzymes may exist in humans, opening the door for promising applications to improve human health. Published in the journal Cell Host and Microbe on June 18, 2025, this study involved multiple co-authors from UC San Diego and other institutions. It highlights the potential for targeted microbial therapies to treat obesity, diabetes, and other metabolic diseases by leveraging time-sensitive microbial functions. Future research will explore engineered bacteria in models of obesity and diabetes to confirm these metabolic benefits. A key discovery in the research was the role of bile salt hydrolase, or BSH, an enzyme that protects metabolic health. Engineered gut bacteria expressing the BSH gene improved body fat, insulin sensitivity, and glucose control in mice. The study revealed that engineered E. coli bacteria producing various BSH variants showed significant metabolic improvements, particularly with the variant mimicking Dubocella newyorkensis, which exhibits a specific daily rhythm. This underscores the importance of not only what we eat, but also when we eat, emphasizing that the timing of food intake is critical for metabolic health. This has been your daily brief in science. To read more about these stories, follow the links in the episode bio. You can also subscribe to these updates via email at www.brief.news. For more daily podcasts about the topics you love, visit brief.news/podcasts. Tune in tomorrow. We'll be back with everything you need to know.